Welcome to the webinar. I'll do an abbreviated version. Today's topic is biological and microbial aspects of septic system pollution. So we'd love to offer the continuing education credits for this. You must be registered for the webinar and attend for a minimum of 60 minutes. So we look at the logs for that, and that's a state requirement. And we'd be pleased to offer you those continuing education credits. Going to the next slide. Today's speaker, we're going to start with the Office of Research and Development speaker this month. Dr. Orrin Shanks is a geneticist whose primary specialty is the application of molecular technologies for environmental microbiology. He has been with the EPA for about 10 years, working with the Office of Research and Development and the Water Supply Water Resources Division. Dr. Shanks has received his undergraduate and master's degree from the University of Wyoming and his PhD from Oregon State. So with that, it's thundering quite a bit here, storming in Cincinnati. If you hear thunder, that's what it is, and it's quite appropriate for today's topic. So if we can go to the first slide, Dr. Shanks, you can take it right over. And sorry for the snafu with the audio to begin.
Okay, thank you, Tom. So my name's Orrin Shanks, and I'm a geneticist with the Office of Research and Development. And today I was asked to do a presentation on microbial source tracking of septic system pollution in receiving waters. The talk that I prepared today can be organized into 4 parts. I'm going to start with some background information on fecal pollution in the United States, what are EPA's responsibilities for fecal pollution, what types of microbial source tracking tools are available, and what does microbial source tracking mean, things along those lines. For the second part of the talk, I'm going to tell you about Office of Research and Development and Office of Water efforts to develop human microbial source tracking methods that are standardized. These methods then could be used out in the field to characterize septic pollution in surface waters, for example. The third part of the talk, I'm going to tell you about 2 different studies that have gone on here at ORD, one that's completed and one that's in progress that have to do with septic pollution. And then finally, I'm going to provide some additional information on outreach and resources that are available to the people out there that want to learn more about these different types of technologies and how you might be able to implement them in your watersheds of interest. So fecal pollution is a nationwide problem. There are several organizations, including the EPA, that have shown that fecal microbes are the most common biological contaminant in surface waters. There's been studies done on estimating the amount of fecal material produced on a yearly basis, and one of these estimates done by the USDA estimates over 1 billion tons of fecal waste are produced just from poultry, cattle, swine, and human. So when you think about other sources such as wildlife not being factored into this, you can start to realize that this is an enormously large amount of waste being produced each year that requires management and definitely has the ability to get out into the environment and cause some situations. Now, the EPA has a number of responsibilities when it comes to fecal pollution. One of the main ones is to protect and restore waters for recreational use under the Clean Water Act. But we also are required to develop new or revised ambient water quality criteria on a regular basis for the risk assessment of beach contaminants, and this is under the Beach Act. We also facilitate and run a number of programs that are designed to help with management of point and non-point pollution sources, such as total maximum daily load programs, national pollutant discharge elimination system programs, and national estuary programs. So the source of fecal pollution is very important. Currently in the U.S., the U.S. EPA recommends fecal indicator methods that do not discriminate between different pollution sources. So one way that we refer to that is a general fecal indicator, meaning that these are things such as E. coli or enterococci that you might grab a water sample and then culture and count And this gives you information about the total fecal load in a sample. But one thing that we know is that different sources of fecal pollution can pose different public and ecological health risks. And most water sources are impaired by more than one source. And because of these reasons, it becomes clear that having information about particular sources of pollution, in particular human fecal pollution that could come from septic systems, for example, can be very important and useful for water quality management. So because general fecal indicators measure the total load from a number of different animal sources, scientists many years ago came up with the concept of microbial source tracking. And what this means is that these are methods that are designed to collect, isolate, identify, and measure what I'm going to call a host-associated indicator from an environmental sample. So you can see that I've colored, bolded, and highlighted host-associated to really emphasize this point. So this is in contrast to a general fecal indicator, which is what I talked about on the other slide. So this is a host-associated indicator, meaning that when you measure this indicator in the water, it tells you something about a particular animal group or pollution source. And what I've done here is kind of made a flow diagram that shows how these methods work on a very overview type level. So you'll see here on the left you have a body of water that you believe to be impaired. You would collect a sample, concentrate or filter that water sample. You would use some type of technology to measure that host-associated indicator. It gives you data that then can be interpreted to help to identify a particular source of fecal pollution. In this case, we're saying septic pollution. And that this information then can be used by water quality managers to make decisions that impact the safety of the public and of the and as well as the ecology of the watershed or beach of interest. So diving a little bit deeper into microbial source tracking methods, there are a number of ideal attributes for these methods, or characteristics or qualities that are needed in order for them to be useful. What I've done here in this table is listed some of these different attributes, and I want to take a few moments to go through them so that everybody can get an appreciation of what is expected and what's needed from a microbial source tracking tool. So the first thing that I wanted to point out is something called host specificity. And this is what I was referring to in the other slide, where the indicator needs to be closely associated with a target population. So for example, if you wanted to identify septic system pollution in your surface waters, then you would want to use a method that's closely associated with human populations. A second attribute of a good human microbial source tracking method then would be that we know the host distribution of that indicator. And what I mean by that is that we understand the frequency and concentration of that indicator in the target population, in the example here, humans, but also in non-target species if it does exist. Third, it would be ideal if it was a quantitative technology, meaning that that we can measure directly from a water sample the indicator concentration, and that this could be done in an accurate and reproducible manner. Another thing that would be really useful is what I like to refer to as expert consensus. So there are a number of options out there for microbial source tracking methods, and one of the bigger challenges as an end user is how to decide which method to use and when. So it would be wonderful if there was an expert consensus or agreement among the majority of professional researchers on particular sets of methods and when they should be used. Another important attribute would be method standardization. And so what I mean by that is that there would be a standard operating procedure with uniform data acceptance criteria so that the method could be implemented in the same way across laboratories. The method should also be validated through a multiple laboratory study and shown that it adequately meets application needs. And then finally, I think that it's very important that there are clear field demonstrations showing comprehensive real-world examples of how you use these methods to solve specific problems out in the environment. So this is a tall order, and for the last, the 10 years that I've been here, this has been the big part of the work that's been done here in our group, trying to deliver human technologies that can do these things. So you can imagine that if we had technologies like that, we could use them to help solve a lot of different problems out in the environment. We could use them to identify non-point pollution sources. We could use them to help with wet and dry weather risk assessments. We could use these types of tools to determine eligibility of a site for alternative recreational water criteria. We could do pollution impact assessments. You could use these for in cities where there's combined sewer overflow consent decrees to show that the impact of the work you're doing is having a positive influence on water quality. You could use these tools to prioritize sites that are impaired for remediation when there's limited funding. And you could also use them to evaluate other types of best management practices that might be implemented in a community, green infrastructure types of things, or storm water flow situations, or even agricultural situations. Okay, so now we're going to switch gears. I've gone through some of the background now, so we should have a good idea of what a general fecal indicator is, what microbial source tracking is, what kinds of attributes are we wanting in a good method, and how these methods could be used to help solve problems in the water quality management arena. Now I want to tell you guys specifically about efforts that ORD and Office of Water have been doing to provide standardized human microbial source tracking methods. And these methods would be ideal for characterizing septic system pollution, for example, in surface waters. But before I get into what we're doing, I want to just do a little review of what I call the anatomy of an MST method. And so if you look here on the right, I have a flow chart that shows different steps or different parts of a method. So when I say a method, I'm talking about sample collection, sample preparation, the actual indicator measurement, as well as the data analysis interpretation. So it's kind of like this holistic idea of what a method is. And the point that I want to make is that if you alter steps in these methods, it can change the performance. And so hence, it becomes really important that a method is standardized if it's going to be applied across laboratories and used in some type of regulatory or policy environment. And so when I say method standardization, what I'm talking about is a formal method protocol. And as I just said, that this is necessary for widespread adoption. So this next slide here gives you an overview of the approach that Office of Research and Development and Office of Water have used to develop a human MST method, a standardized set of methods. And this is just an overview. The next few slides will go into some more detail on each one of these steps. First one is method selection. So how do we decide which detection technology to use, which indicator should we use out of all the ones that are available? Once we make these decisions, we need to do a technical evaluation of these methods that's peer-reviewed. We need to follow this with a method validation where we establish a standard operating procedure, we establish data acceptance criteria, and we do this through a multiple laboratory study. And once it makes it through all of these steps, it can be published as an EPA method, making it eligible for use in future EPA policies and programs. So there are a number of detection technologies. So people on the call that are familiar with microbial source tracking know that there are many different methods out there, many different tools and different technologies. And so this is just a list of some of the ones that are currently in use. You have things such as microarrays, deep sequencing, different types of PCR and quantitative PCR, antibiotic resistance profiling. There's different types of chemicals that can be measured. And there are even groups out there using canine scent detection. So how do we figure out which method we should choose to standardize? Well, the way we decided to do this was through expert consensus. So what we did was we teamed up with the Southern California Coastal Water Research Project Authority out in Southern California, who was conducting what they call a Source Identification Protocol Project, or SIP. So EPA, along with SQUIRP and 3 other organizations, Stanford, UCLA, and UC Santa Barbara, designed a study that was basically a blinded study that was a public challenge to expert laboratories to take their methods of preference and see how well they perform with a blinded set of samples. And what ended up happening is we were able to recruit 27 different expert laboratories for this study using 41 different microbial source tracking methods, including everything that I showed you in the last slide. So we had the canine scent, the dogs there, all the way through sequencing and PCR involved. This work actually resulted in 12 peer-reviewed manuscripts that came out in a special issue of Water Research in December of 2013. And so what I'm going to do now is just highlight on some of the key findings that influenced our method selection for standardization. The first thing we saw was that the majority of the experts, over 90%, favor a PCR-based technology. And when I go through some more of these slides, you're going to see that there's references below them. So this is just here for people that want to, after the talk, go back and learn more or get more information about a particular study. The second thing I wanted to point out is that qPCR methods were highly reproducible across labs, but only when the protocol was standardized. And the other thing I want to point out is that they were able to identify the top human-associated qPCR methods. And so what we were able to use this information through a process of expert consensus to arrive at which technologies and which indicators we should focus on for method standardization. The second step is technical evaluation. So a group of government and academic researchers got together and performed a rigorous assessment of the performance of these methods that was subject to peer review through the publication in a scientific journal. We made sure that we adhered to the minimum information for publication of qPCR experiments. So people on the call that are familiar with qPCR probably have heard of these MICE guidelines. And these are basically a set of guidelines saying that this is the minimum information and controls and practices you need to do to be able to generate high-quality data that's defensible. Okay. And then the last thing we wanted to do was we wanted to incorporate and optimize using newer reagent systems that have been designed specifically for environmental samples. And so if you want to learn more about this, a postdoc from our group, Hyatt Green, was very instrumental on this, and we published a paper in 2014 on that. So the next step then is a multiple laboratory validation study. So the Office of Water, the Office of Research Development, and the Environmental and Innovation Technology Cluster teamed together to conduct a multiple laboratory study focusing on 2 human-associated qPCR methods. There were 14 laboratory participants, and all the laboratory work has been done, all the data analysis has been done, and there are now drafts of these EPA methods that are being reviewed internally by the EPA, and hopefully they're going to be published sometime in the next 12 months. And everybody's excited about that, and I'm happy to answer more questions that people might have after the presentation. Okay, so now we're going to go into the third part of the talk, which is talking about some of the septic pollution research activities that we've been involved in here in ORD. The first one I want to bring your attention to is a question that many of us have out in our watersheds. Does human fecal pollution originate from leaky sewer lines or a failing septic system in my watershed of interest? And so this research I'm going to show you was done with a postdoc in our lab named Lindsay Peed, and in collaboration with Dr. Christopher Nietzsche of the EPA. And what we did was we went out into the East Fork Little Miami watershed. It's in southeastern Ohio. In this area, there's a range of septic and sewer use intensities. And we decided to sample 9 catchment areas using small stream sampling approach over a 2-year period. And with these water samples, we measured general fecal indicators as well as 3 different human-associated qPCR methods. And based on the general indicators, we know that these waters were unsafe for recreational activity over 40% of the time. So there's definitely fecal pollution there. For more information on this, you can see down here the publication from Lindsay. So another aspect of this study was to incorporate GIS mapping. And so what I have here are 2 panels. In the left panel, you can see one of the catchment areas, and we've layered septic sewer line information on top of the catchment area. On the right one, we've layered septic system locations. And what this allows us to do then is to calculate the area of this catchment and normalize and come up with a density of sewer lines or septic densities. And when you do this across all 9 catchment areas, what you end up with is a gradient of sewer and septic use. And so you can see this here in this GIS image. We have the 9 catchment areas, and they're color-coded with this light pink, meaning 0 septic systems, all the way up to almost 90 septic systems per square kilometer. So when we look at, when we organize the watershed into these 9 catchments and construct this gradient of use between sewer and septic, what we found was that there's a negative correlation between septic and sewer densities, which intuitively makes sense. But what this allows us to do now is we can now go and do microbial source tracking on the water samples from these catchment areas, apply it to this catchment gradient, and ask the question, does human pollution trend with sewage densities, septic densities, both, or neither? Okay, and when we did this, what we found was that human fecal pollution increased with septic density, but that it only did this during wet weather sampling. So in this plot here, we have a scatter plot, and on the x-axis, we have the log septic density, ranging from 0 up to almost 90. And then on the y-axis, we have one of the microbial source tracking markers, the copies per reaction. And you can see that the points are color-coded by whether it was a wet weather sampling event or a dry weather sampling event. Up here on the right, we have 3 different human MST methods showing the correlation coefficient and the significance of the correlation. And so what we saw here was agreement among 3 different human source tracking markers that during wet weather, the higher the septic density was, the more human fecal pollution there was. And because there was a negative correlation between sewer line and septic, the opposite is true, meaning that the more sewer lines there was, the less human pollution there was. So what this tells us in this watershed is that the human fecal pollution portion of that's causing the impairment in this watershed is happening after rain events due to septic system presence. And the second study I want to tell you about has to do with the composition and decay of human fecal pollution sources. So today we're talking mainly about septic system pollution, but there are other types, sewage, treated, untreated sewage, and even raw fecal material that have different ways of finding their ways into surface waters. And so we wanted to do a study to ask, does human sewage, septic, and fecal waste display similar decay trends in a marine water environment? And so we did this by trying to mimic the release of pollution into surface waters using a submersible mesocosm device. And so I want to point out that this work is being done by a postdoc that's currently in our lab named Pauline Wanchugi, and that this submersible mesocosm was designed by 2 EPA scientists here, Asia Karagic and Brian McMinn. And basically what we're doing is we're trying to mimic the release of pollution, and then we are studying the influence of different types of environmental stressors. So in other words, are they exposed to sunlight or not, and other types of environmental stressors. And then we're measuring a panel of indicators general indicators through cultivation, through molecular methods, bacterial and viral-based, as well as microbial source tracking methods to see if all types of pollution and indicators behave in the same way or in different ways, because this can be important for future applications. So ending up the talk here, I want to provide some information about different outreach and resource opportunities. There are periodically regional and state workshops that go into much more depth on microbial source tracking. I had already mentioned earlier about water research, MST special issue. That's a great issue to pick up and kind of get a current state of the science of where things are at from a large recent study that was done. There's also a document that has been published by Region 10, EPA Region 10 laboratories on microbial source tracking to support TMDL development. ORD has also published a microbial source tracking guide document in 2005, thanks to efforts of Jorge Santo Domingo. And then finally, we have standardized EPA methods for human fecal pollution characterization, hopefully coming soon. And we're all excited about that and hoping that this is going to promote the use of these methods in a more accurate and reproducible way. And before we move on to the next part of the seminar, I want to definitely take a moment to acknowledge the different teams of researchers that were involved in this work. So this was an enormous amount of work that took many years to do. And I've already mentioned a number of the postdocs by name. I do want to mention two other individuals from the EPA research team, Kathy Kelty and Mano Savigins, both of them were involved in every study that I showed you today. And then the other thing I wanted to do was thank the 14 laboratories that volunteered their time to participate in the multiple validation study. So those organizations are listed on the right here, along with Thermo Fisher, who allowed for use of qPCR instruments for a number of these laboratories, and the EPA Environmental Technology and Innovation Cluster, which funded a lot of the work. So with that, I'm ready to step down and do whatever's next.
Okay, great. Thanks, Oren. And excellent talk, and I apologize for our audio snafu, and I apologize to the audience, but outstanding talk. One note before we switch over to our second speaker is, and I'm not sure if we covered this in the preliminary work, is that please put in the number of people in your room and put it into the question box on the little dialog box off on your screen. If we could have the number of people in your room, it helps us show how important this seminar is. Also, if they're interested, if they're not registered but they're in your room, please verify their names for us, and would be able to send them a CEU credit certificate also. So we can do it that way also. Oren, we've got a few talks, a few questions for Oren. Also, if you have only one person in your room, you don't need to bother sending in the number, only if there are multiple people. So if you have any questions for Oren, feel free to put them in the question box. We'll organize them until after our second speaker. And with that, we'll start right to our second speaker, who is Alfonso Blanco. He is an environmental engineer with 40 years of domestic and international experience on wastewater projects. Presently, he is working for EPA's Office of Wastewater Management in the Sustainable Communities branch, where he's worked there for 20 years now. He has a BS in Civil Engineering from Merrimack College, an MS in Environmental Engineering from Tufts University, and his PhD is in Remote Sensing from George Mason University. Dr. Blanco, I'll give the stage to you.
Good afternoon, everyone. My name is Alfonso Blanco, and again, I work in the Office of Wastewater Management, the Centralized Wastewater Group. I'll be presenting a demonstration project by the name of Table Rock Lake, which is located in the Ozarks region in the southwest part of Missouri. This presentation will cover several advanced on-site wastewater treatment technologies, as well as disposal systems in difficult conditions. The structure of the presentation basically is an introduction, which shows the study area, what the statement of the problem is, objectives, some background. Then methods and materials, the selection of the sites, and the treatment system, and also monitoring results, monitoring data, and also the cost related to each individual system. And finally, lessons learned, and that will cover the responsible management entity that's basically in the voluntary national guidelines for on-site wastewater treatment systems. Table Rock Lake is located in the Ozarks region in southwestern Missouri, which is an important tourist area. They receive about 30 to 40 million visitors annually. The area is characterized with steep slopes, fractured limestone, bedrock, and porous soil. So very difficult conditions for a septic system. This area is a resort area with many failing septic systems around the lake. It covers about 43,000 acres, which includes the James River watershed and Stone County. The lake back in 2002 and before then was on the 303D list of impaired waters for nutrient, eutrophication, biological indicators, chlorophyll, and nitrogen. Give you a little background information. Table Rock Lake Water Quality, Inc. is the grantee for this project, which received $1.94 million grant from EPA back in 2002, with a grantee share of $667,000. In 2004, a responsible management entity named Ozarks Clean Water Company was established to operate and maintain the advanced wastewater treatment system. The objectives of this project were to demonstrate the different types of advanced wastewater treatment system available, and also removing BOD, total suspended solids, ammonia, phosphorus, and fecal coliform. Another objective was to develop a management program following EPA's recommended management models in the voluntary national guidelines. Third objective was to identify and address legal impediments for the widespread adoption of this system by changing the regulatory bodies and wastewater industry perception of these systems in order to be accepted in the state. What's the problem? The problem was a septic system leaked through the fractured rock and soil and entered the lake, as you can see in the photo, which generated high levels of nitrogen and phosphorus. Consequently, this impacted the water quality by creating algae biomass. One major problem was that the population grew around the lake, and along with that, more septic system failures. The existing systems were not upgraded to compensate for the increase in wastewater volume. This is a typical diagram of a septic system, which consists of a septic tank and a drain field. Wastewater enters the tank, gets primarily treated, gets discharged to a drain field, and is finally treated by the soil. There were 24 sites that were selected, consisting in clusters, residents, restaurants, but only 4 sites were monitored. A need survey was done to determine which sites will be selected for the project. That means that the authority went and talked with the individual homeowners to see if that was acceptable to them, and also discussed environmental needs, feasibility of installation, potential for cost sharing, and the honest interest in the project. So they were also looking for fees to supplement that local matching funds. The types of advanced on-site wastewater treatment system, there are 4 of them. Constructed wetlands, the aeration fixed film media filters with foam cubes or peat moss, and recirculating sand filters. Final disposal will consist of drip irrigation with imported soils. The in-situ soils were not adequate to treat the effluent. Constructed wetlands. These are national wastewater treatment systems. The components are basically septic tank, a settling tank. It's got an impermeable liner. It's got a gravel substrate, mulch, and water-loving plants, a distribution system, collection system, and water level control device. And the plants basically provide for the aerobic treatment. The treatment of the effluent is disinfected by chlorination or UV. Some of the advantages and disadvantages. And the advantages, basically, the is a low cost for construction, which is about 60% less than conventional systems, and O&M. It's beneficial for wildlife and very aesthetically pleasing. However, it requires a large surface area, and it's not recommended for seasonal residences. Maybe the best systems for nitrogen removal is this constructed wetlands. The system is ideal for failed conventional absorption fields or low absorption and percolation. First system, this is aeration with fixed media, which basically the two compartments to it. One is the primary zone, which is a settling zone, and then the secondary zone where it has a mechanical aerator, which is accessible for service or replacement. The system is installed underground except for the blower, so aesthetically it's very appealing. It generally requires the same land area as conventional septic systems. However, the disadvantage to this system is that typically you have higher cost than the conventional septic system. It requires more maintenance than the conventional ones due to mechanical and electrical components. Blower could be relatively noisy, so it may need to be enclosed. This again is the two compartments in the septic tanks with an aerated chamber and a fixed film media. Pollutants are treated by bacteria, and the units can be retrofitted to prevent failures, increase the treatment values, protect the environment, and increase the property value. This aeration fixed film media advantages are that basically it's easy to pump, it has the same land area as conventional, it's got a small mechanical aerator, and it's well accepted by the public. However, it's more expensive and requires more maintenance. Also needs backup power and a blower enclosure. Media filters with foam cubes. Basically, this system consists of a septic tank and a separate tank which contains the foam cubes. Essentially, this is a biofilter system when the effluent is sprayed over the foam filter. Dosage is about 1/10 of a gallon to about 1 gallon per cubic feet, and it can be used either in a single pass or on a recirculating system. These are the advantages and disadvantages. Basically, are applicable to small lots, and it's easy to maintain. However, spray nozzles tend to become clogged, which will distribute the effluent unevenly. Another system, media filters with peat moss. The wastewater effluent from the septic tank is pumped to the fiberglass shell containing the peat moss. And naturally occurring fungus and bacteria treats the wastewater. This is basically an attached growth pre-treatment system consisting of a fiberglass tank with the peat moss. The wastewater is discharged to the soil through a lateral or a drip irrigation system. The advantage is that this is a gravity system. It's got low O&M with good removal rates. However, the peat moss requires disposal and needs to be replaced every 10 to 15 years since it gets clogged. And one of the problems is a lot of landfills, disposal areas will not accept the peat moss that's been contaminated. The circulating sand filter, this is a system, consists of an aerobic fixed film biofilter system, which will remove the solids, and then chemical absorption will remove the nutrients. The system basically splits the flow going to a dosing tank and the soil. And the components are a dosing tank, pump, and controls, distribution pipe, the sand filter bed with a drain system and a return line. System provides about 95% removal of BOD and total suspended solids. The advantages are ease of pumping and maintenance, simplicity of operation, again 95% BOD and TSS removal, 50% nitrogen removal. However, it requires a larger surface area, which is more expensive than a conventional system. The drip irrigation with imported soil, and some of the system in particular consists of a half-inch plastic tubing and drip emitters placed about 2 foot apart and 6 to 9 inches below the surface. There is a holding tank that collects the treated wastewater, doses the drip field using a pump and a timer. Many situations, this is the only technology that will work. It's easy to design and install. It has a low system profile that can operate in sloping sites, relatively high preservation of landscape features, and can accommodate marginal soil sites. However, the disadvantage is that it's expensive, and the maintenance of the drip irrigation system requires additional considerations such as a good vegetative cover and regular monitoring of the dispersal and aeration components. Backflushing of the tubing is necessary to clear out solids deposited in the lines. And also pressure release valves may be necessary to lessen freezing and infiltration of soil in the drip emitters. Monitoring, I mentioned before, 4 sites were monitored. We use a half-pipe gravity lysimeter, which were installed on 3 sites with the drip irrigation into the imported soil, and also piezometers were installed on 1 site. The samples were taken from the septic tank effluent and treatment effluent, pre-treated filter effluent, the effluent that has passed through the soil. So these gravity lysimeters collect treated wastewater as it percolates through the saturated soils, and the piezometers, as you can see, are vertical pipes inserted into the soil at varying depth. At this site, with imported soil, the soil depth was 12 inches above the drip tubing. Approximate depth between the drip tubing and the lysimeter were from 1 to 9 inches. And it was monitored for about 20 months. This is a view, cutaway view of the half-pipe lysimeter and the piezometer being installed. These are some of the monitoring results for the 4 sites. As you can see, for the different systems, the constructed wetlands had the best performance, and including ammonia and phosphorus. The media filter using the foam using the foam cubes also did fairly well. 92% fecal coliform removal. One mention here is ammonia is regulated in Missouri, but total nitrogen, nitrate, and nitrates are not. Additionally, the phosphorus rather than nitrogen was the limiting nutrient for this project. And this is some of the cost. And as you can see, the constructed wetlands are the most expensive one from all of them. And as you can see, some of the manufacturers are in the column which shows the estimated technology. And this is EPA's determined that in order to provide a high-quality wastewater treatment, we have to look at proper maintenance. EPA's voluntary national guidelines for management of on-site and cluster decentralized wastewater treatment system outlines 5 management models for providing a guide to match the needed management controls with public health and water quality. These models are in order of increasing management controls. I'm going to discuss basically 2 of the models, which is Model 3 and Model 5. Now, we're getting to what basically we learned from this project. And one of the biggest results the state and county regulatory agency and installers of advanced wastewater treatment systems accept as a solution to failing conventional septic system and the use of drip irrigation with imported soil. This project provided the scientific information and experience needed to require advanced on-site wastewater treatment system and drip irrigation in tough site conditions. Local regulatory agency changed the wastewater ordinance to require renewable operating permits for advanced on-site wastewater treatment system, which is that EPA management Model 3. In the past, this system was not widely accepted as feasible or practical, and contractors did not want to install them. So this project provided regulatory agencies with the necessary scientific evidence to accept this advanced wastewater treatment system as a standard rather than experimental. RMEs removed the maintenance responsibility from the developers and the homeowners. The formation of a responsible management entity, Model 5, was the Ozarks Clean Water Company, which was established in 2004. Now, another lessons learned is this renewable operating permits, EPA management Model 3, basically changed the public perception on advanced on-site wastewater treatment system, which was accepted as a solution for wastewater treatment. And now local regulatory agencies now requires renewable operating permits for advanced on-site wastewater treatment systems. This is Model 5, and this is some information on Ozarks Clean Water Company, which is a not-for-profit company. It owns about 15 small decentralized wastewater plants, and it has about 400 service members. The EPA management Model 5 owns, operates, manages a decentralized wastewater treatment system in a manner analogous to central sewage. RME ownership is recommended where new high-density development is proposed in the vicinity of receiving waters. And states may need to establish a regulatory structure to oversee the rate structures that RMEs develop. User fees cover all the necessary O&M and repair work. The initial fee for a single-family house was $28.53 per month. Now, the current average monthly service fee rate is $32 per month. These are some of the examples of RMEs to operate, maintain, repair system to comply with performance criteria, which are stipulated in the operating permits. Submit compliance monitoring reports to regulatory authority according to schedule stipulated in the operating permits. Hire certified, licensed evaluator, designer, operators, and inspectors maintain system monitoring and service records. And this is some of the references that I use to prepare. Actually, there is a paper which is going to be sent out with the slides. And there is a very detailed report, which is the first reference that you see, okay? And that's where most of the information was taken core. I want to thank some of the people that worked with me on this project, with Gopala Borshett, Executive Director for Table Rock Lake Water Quality, Inc., and Dave Castelletto, Executive Director of Ozarks Water Wash, and my intern, Allison Martin. Thank you very much, and here's my contact information at the bottom. Thank you very much.
Well, thank you, Alfonso. So we're done with our two speakers. It's 3 o'clock and Eastern time almost. If you have to go at this time, we understand. But there is time for some questions. We're here until, for the interim, maybe up to 3:30 if need be, to answer questions. So let's start some questions with Dr. Shanks. So the first question is a question regarding the SOPs for previously described method. Have you developed or are developing an inter-laboratory quality control program?
Okay, thanks. I guess everybody can hear me. Okay, yeah. Great question. So one of the things that we're trying to do with this SOP, because we have multiple laboratory data from 14 different labs, one of the things that we did was we developed performance benchmark and data acceptance criteria. This will be part of the SOP. It'll also be written up as a formal publication that will undergo peer review and hopefully be published. But great question. We're not going to be able to implement a program, but what we can do is we can provide directions and the procedure and recommended in the EPA method protocol. So what you're going to find in there is a procedure with data acceptance thresholds for different steps in the method that will allow you to compare how you are performing in your laboratory compared to how the people in the study performed. And then this will give you a good idea and of how well you're implementing the method, and it will also allow you to troubleshoot if you are having problems, where you're having those issues. Does that come?
Yeah.
Okay.
Well, thank you. Thank you, Ron. Another question for you, Ron. What are some of the best performing MST methods for identifying both animal and human-specific host sources of pollution?
Another great question. I get asked this question a lot, and I wish there was an easy answer here. One of the things that's the nature of microbial source tracking is that these are bacteria or indicators or gene targets within bacteria that are shed from animals, and so it's a little transient, if you will. But what I can tell you is, and because of that, what we've actually put into the EPA method is a protocol on recommending that you go out and collect reference fecal samples in your area of interest, and then test the different methods to ensure, to calibrate, or however you want to say it, so that you're making sure that you're using the right method for your area. So in the SIP study and in our experience, some of the better performing human methods are the HF183 method, the HUM-M2 method, the BAC-H method is good, BAC-HUM is a good method. For ruminant methods, there's the BAC-R method, or the RUM2-BAC method is a very good method. For methods that are specific for cattle, we use the CAL-M2 and CAL-M3 methods a lot. For swine, the method that we prefer is called PIG2-BAC. And then there are also some avian methods that are good, and some dog methods as well. Some of the better avian methods are the GAL2 qPCR assay and the GFD qPCR assay developed by Hyatt Green and Kate Field at Oregon State. So there's some information there. I'm happy through email or whatever to try to provide more detailed information if needed.
Okay, great, Oren. One note on your screen, there's the schedule for the next talks. One comment is that the August talk has been moved up 1 week to August 18th. So that's to note if you have an old schedule on your bulletin board or such for those that need to go. Here's a question for Alfonso. If you're ready, is the O&M tracked in the database?
I believe the Table Rock Lake Water Quality Inc. will have that information.
Great, thank you. Here's another one for you, Alfonso. Why wasn't nitrogen removal rates shown on AWT's results page?
Well, as I mentioned, basically ammonia is regulated in Missouri, but the total nitrogen and nitrates are not.
Okay, super. Here's one for Oren. When do you expect publication of the results of the decay trends in the marine environment study?
Great question. So we've completed the study, we've completed the data analysis, and we're writing the manuscript right now. But you know, it's kind of at the mercy of the peer reviewers and the journal as to exactly when it would become and if it would be accepted and so on and so forth. But it's our hope that there might be something out there in as little as 6 months from now.
Okay, super. Here's one for you, Oren. Have you considered the age of the septic systems as another factor?
No, we haven't at this point. You know, the study that I showed you in the East Fork watershed was basically just looking at, you know, septic densities and water quality in these small streams. So, you know, there's a lot of great things that I think we could do as a next step in that study, you know, to go out there and see how many of the systems actually are not performing correctly, you know, how deep are they buried, where are they located, doing some more precision sampling during storm events to get a better idea of exactly what's going on there. But great question. Thanks.
Okay, Alfonso, here's a question for you. What brand of drip technology was used? And the question is whether it was American.
I believe it was, the name is naphthalene. It's a purple tubing.
Okay.
And that's a manufacturer.
Okay. Here's another one. What was the public perception during the establishment of the RME as you went from individual O&M to the RME concept?
That is a question that I don't have an answer. I think the Table Rock Lake Water Quality Inc. is the ones that would have, you know, they can respond to that.
Okay. Here's a question. Oh, that's quite all right. Here's a question for Oren. If septic systems are a minute percentage of the risk compared to animal livestock, shouldn't it be more effective to address that?
Okay, so the way I would answer this is I imagine this question is coming from one of the first slides I showed where there was showing the proportion of fecal material produced from different animal sources. And you could see in that one that actually when it came to swine, poultry, and cattle and human, the human actually on a mass level, it represents less than 1/100% of the mass production per year. But in terms of risk, it represents the largest portion for humans. I think there was a miscommunication on our part here because in terms of risk, the human pollution sources is perceived to be the highest risk for public exposure. And then that's why the emphasis is on that pollution source to start out with instead of us doing all of this, for example, with cattle pollution or another animal source. So I hope that answers the question.
Okay, great. Here's a question for Alfonso. are these peat systems actually using peat moss or peat itself?
Peat moss.
Okay. And here's a question for Oren. What about the qPCR digital DNA testing machines and protocol?
Okay, so there are different types of qPCR for people that aren't familiar with these technologies, and there's a new type of approach that's just recently become available in the last few years called digital PCR. And basically, for a little bit of technical information there, is that it uses a most probable number approach to quantify data. And we're starting to see research laboratories use this technology for source tracking and for general fecal indicator measurements in water samples. It advertises that it could have some advantages over other approaches in terms of dealing with inhibition, in terms of limits of quantification and things along those lines. But what I would say at this point is that there are very few papers that have actually, peer-reviewed papers that have used this technology for these particular applications. As a matter of fact, I may only know of one or two to date. So in my mind, it's a new technology. I do think it has a lot of promise, but I think it's more kind of in the research phase right now to understand if all the advertising is true. I do think that to give you an idea, we have just obtained one of these instruments ourselves, and so we are looking into that as well. And it seems like a great technology, and hopefully it'll be used more in the future. I guess when I talk about these types of technologies, I like to refer to them as emerging technologies. So things like microarrays, next generation sequencing, as well as digital PCR. In my mind, these are emerging technologies that we need to always keep one eye to the horizon and trying to improve and implement new technologies as they come on board. But for right now, I would recommend, if you had to choose between one or the other, I would choose the qPCR just because there's hundreds of publications with these, using these, types of technologies that have been peer-reviewed and substantiated, where with the digital PCR it's still very new. And it may end up working better, but at this point we don't know, we don't have a lot of information to convince us one way or the other.
Okay, just on somewhat of a related question, what is the status of standardizing EPA's method? Is there a formal program being organized?
So yeah, well, there's not a formal program, but what I can tell you is, so the process of creating an EPA method is really a good example of Office of Water and Office of Research and Development working together. So it starts out with administering the laboratory phase of the test. And this is when test materials go out to the volunteer laboratories and they generate their data. So all of that is complete. All the data has been submitted to ORD. It's all been analyzed, quality controlled, written up. Drafts of the SOPs have been provided along with all of the data to Office of Water. Office of Water now is doing their formal independent review of ORD. And so that's where it's at. And it's our hope that it'll be published by the end of this calendar year. But again, I don't want to put, put anything in stone at this point. I think if you, in the last slide I had there, I say coming soon, hopefully within the next 12 months or less.
Okay, great. Thanks, Mark. Alfonso, here's a question for you. Why was the O&M on the constructed wetland so high?
Good question. I think in the technical report, it would show why, what components, the cost for each of the components. We're talking land, and you're talking the septic tank system, and and pumps, and dispersal. So it is be more expensive than to operate and maintain. However, you get better removals from the constructed wetlands rather than the individual systems.
Okay, thank you.
Does that answer the question?
Sure. Okay, here's a question. Here's another question. How do you propose to transfer the technology into the private sector? What's the cost of installation, treatment, and operation cost? What is the market size, and who is the client for this technology?
Well, I'm going to try to answer. I think that's a question for the Table Rock Lake Water Quality Authority.
Okay.
I don't know the answer.
Okay. Here's a question for Oren. Can you comment on the limitation of the PCR-based assays related to inability to discriminate live from dead organisms? Why did this not cause experts to lean toward a method that can discriminate?
Well, good question. So, one of the things that I didn't get into in this talk is that I guess we did a little bit. There was a slide where I showed many different types of problems that could be solved with microbial source tracking. And so my response to your question would be that for a lot of applications for microbial source tracking, it isn't necessary that the cells are live or dead, but certainly for other ones it could be. So, if you were trying to predict health risk exposures with a human source tracking marker, then one might argue that it would be better to have a measurement of the live cells versus the dead cells. But again, this could go either way. So my easy way out on this question is to say it really depends on what the application is that you're trying to use. And then I might even argue that for the applications where people would argue that you would need this information, that there might be ways to estimate health risk without having to worry about that. But good question.
Okay. Here's one for Alfonso. What was the original waste strength of the 4 sites? Alfonso, you still there?
I'm sorry, we're on mute. I have in the paper that we're going to send out, it has the monitoring results with the influent and the effluent. I don't know, we have several systems here, so I don't know exactly, the BOD, total suspended solids, ammonia, phosphorus, fecal. How many? What exactly, for what systems?
Okay. Here's
several systems.
Did the data collected during the MST efforts consider the possible contributions of fecal by animal sources, which could also be expected to increase in surface waters during or following rainfall periods?
This is a good question. I'm glad that someone asked this, because this exactly highlights the power of a microbial source tracking tool versus a general indicator. So the way I hear this question is, well, there could be other sources of fecal pollution that enter into the waterway during storm events, and undoubtedly there is. And we saw that with our general fecal indicator data as well. But when you use a source tracking method that will only identify the human portion of that contamination, then you can zero in on what's happening just with the human pollution source and not the other one. So if we had tried to do this analysis with a general fecal indicator, which we did not see a pattern between dry and wet weather events, because there's chronic fecal pollution there, but it's from different pollution sources. When we isolate that human signal using a microbial source tracking technology, then out of that indiscernible gunshot of data, there becomes a very identifiable trend. So good question, and that you exactly hit the nail on the head, the heart of how microbial source tracking can give you additional information that you can't get from a general fecal indicator.
Okay, great, thanks, Ron. Alfonso, here's one. I think we haven't covered this one yet. Was the drip tubing metaferm? Alfonso, you there?
Yes.
Was the drip tubing metaferm?
Yes, I believe it was. It was a purple type tubing. That's who the manufacturer was.
Okay, great. Lauren, here's one related to your last one and a previous answer too. Can the first speaker please expand on the risk comment regarding human versus animal waste?
I can a little bit. I mean, again, this is not my exact area of expertise on the risk portion of it, but from my reading and from interacting with other colleagues, what I can tell you is that there are more pathogens, based on literature, that can infect humans that are in human waste versus other animal sources. It's not to say that other animal sources don't also have pathogens that can make us sick. This definitely is the case. There's been some real elegant quantitative microbial risk assessment studies that have been put out there by Jeff Soler and Nick Ashbolt, showing estimates of the risk due to different animal sources. And in those types of studies, it shows that by far human pollution represents the biggest public health risk due to recreational exposure. But in a close second is cattle. And then from there on, I think they looked at swine and they also looked at gulls, and those were much lower. So human seems to be the biggest one, and then cattle a very close second, followed by that. And so, again, this is why we chose to focus on human as our first foray into the world of standardizing MST methods. If we are able to continue this work in standardizing methods, then for sure the second choice would be cattle.
Okay, great. Here's a question about the septic system project, talking about septic systems. So how deep were the septic systems? Is it a groundwater pathogen load or a surface runoff issue?
Yeah. Okay, well, I guess the short answer to that is that we don't know. And what we did in that study is we just measured what was in the surface water on a monthly basis. So I think what that study does do is it gives some empirical evidence that when there is a higher density of septic systems, we see a higher concentration of human fecal pollution. And I think that going back to that study area and doing some more involved research, trying to discern whether or not this is from surface runoff or whether it's from ground flow or interground flow or anything in between would be really interesting.
Okay, and I think this is somewhat related, but there might be something a little different. Oren said that human fecal contamination is perceived to be a greater threat than human health. Does this data exist to confirm this perception? E.g., example, crypto infections in humans usually have a cattle source.
Good question. Again, I'm not an expert, and I'll admit that freely, in medical issues along those types of lines. What I'm familiar with are these QMRA studies that have shown that undoubtedly there are pathogens in other animal sources, zoonotic pathogens that can make people sick. Anyways, but so I wish I could tell you more, but I am fairly confident from what I've read and the collaborators that I have that human is one of the major concern areas, but definitely cattle as well.
Okay. And Alfonso, I think this might be our last question. Are you familiar with the WRF database on assessing costs for decentralized system, individual and community? Is that part of the, is that something you're familiar with?
I'm not. No,
Okay.
I'm sure that
Okay.
I haven't seen it, so I don't know.
Okay. And I think that we're pretty much on time to wrap up. And that thank you for all the questions. Thank you for our speakers, Dr. Shanks and Dr. Blanco. And so that concludes our webinar. Again, the schedule for 2015 is up on the screen. And one date change is August 18th is the new date. Is the new date for August. So please take note of that. And we also have a workshop coming up later this fall in terms of last week in August here in Cincinnati for the Small Systems Workshop, where it's free registration to that workshop. If you can get here to Cincinnati, we'd love to have you. And you can go to the website, EPA's website on Small Systems Workshop, Drinking Water Workshop, and please search for that. And we will get you more information on next month's webinar on that, but please put that in your calendar if you can. So thank you again for this month's webinar. Thank you again to our speakers, and thanks for all the great questions, and hope to see you next month. Thank you very much.